Okay, let's try this again. If you're watching the video, you can fast forward until you see the screen move, in theory. So, for example, my remote's not working. I might be talking to myself here. Bear with me, please. Nah, you know what? Let me take care of you, and then I'll take care of this remote. So yeah, like I was saying, if you're watching the video, we've got quite a ways to go until the lecture actually starts. And what I was going to say is that you can go ahead and fast forward until you see the camera move. However, the camera is not moving. And the noise you hear in the background is me taking taking batteries from one remote and putting it into the other to see if I can get it to work. So yes, bear with me before you fast forward. See if this works. If this doesn't work, I need to go do it manually. Not working. All right. So yes, bear with me. Anyway, the first word for attendance, and this will be, anyway, the first word for attendance is battery, as in the battery I'm working with is not working. So yes, once again, please bear with me. What I'm doing right now, in case you're wondering, is I've turned on the projector so that my screen can be shown against the wall. Once that happens, I can walk over to the camera and manually move it with my hands and point it in the direction it needs to go. But until the screen turns on and projects what I'm looking at, then I have no idea where to move the camera. So once again, please bear with me. Once again, the first word for attendance is battery. So don't fast forward yet, please. Normally, I would just say go ahead and fast forward, but since I'm still working with the camera, can't tell you what to look for. Okay, the projector's on. Let me take a look here. All right, well, it's way too zoomed in. But it is what it is. So let me see. Oh yeah, way too zoomed in. Anyway, it is what it is. There's nothing I can do about it. So yes, I'd say go ahead and fast forward. I guess until you see the PowerPoint show up. So I'll project. Actually, let me go ahead and do it really quickly to show you what I'm talking about. Bear with me, please. 16, 15, 7. There we go. Okay. Excuse me. See, right now it's just showing the projector, but when I share my screen like this, first you'll see this, then you'll see the projector, or excuse me, the PowerPoint, then you'll see me actually start the slideshow, right? So once you see that, that's when you know it's time to stop fast forwarding and start watching and listening to the video. So yeah, go ahead and fast forward until you see the PowerPoint on the screen.
and the parents are too big to crawl through some of the tunnels, so you just hear Preston, and you just hear from another room, Mommy, and you're like, yeah. It's fun.
Well, anyway, the City Museum in St. Louis will attempt to break the Guinness World Record for the most people wearing underwear on their heads.
Well, this is wacky fun.
Yes. This is something I can get behind.
Yeah.
Really?
According to the museum, it's looking to add it to its robust record collection. This includes, the museum has titles for the world's largest pencil, the world's longest seesaw. Couldn't Archimedes or somebody give me a lever long enough and I could lift the world or something like that?
Something along those lines.
And the world's largest tennis racket.
So what underwear would you wear?
I don't know if I
Would you
would.
wear a ladies?
I
Would
don't
you wear
know.
your own?
That's a good. I don't know if I would wear a lady's underwear.
I'd probably go with clean.
Yeah. Good choice.
New. I'd maybe go with the
Yeah.
classic white boxers with hearts.
Oh.
Funny. Yeah.
I think I'd wear girls' underwear if that one girl would call. Anyway, in honor of 314 Day,
That's the area code.
all things St. Louis,
Oh.
the museum is hoping to gather 314 people on March 14th and have them all wear underpants on their head for at least a minute. The record to beat is 270
Maybe we should do this.
right now. Should
Put
we?
underwear on our heads?
Can we
Can we do it Zoom? Or do we
participate?
have to
I
be
think
there?
so.
Virtually,
Oh.
we have to be there.
You'd have to be there.
Yeah,
That's fun.
What is the 14th? 14th
It's a
is
Thursday.
Thursday, definitely a Thursday.
No. Yeah.
This is going to upset all the Pie nerds, 3.14,
Oh,
because that's their
yeah,
take.
that
So
is.
now it's
Oh,
the St.
no.
Louis nerds versus the Pie nerds. This would be really messy.
What did we got? We got May 4th.
May the 4th be
We
with
got
you.
Pi Day. We've got March 4th. That's also a sentence, I guess.
Yeah.
That's what I say. You know, one year I wrote joke on it on the check.
Are you serious?
Then I went, why would I do this?
Good thing they're understaffed right now.
That's the only reason I think I got away with it.
I can't imagine anyone that works there has a great sense of humor. They don't.
Get a load of this. Ha, 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 ha.
Oh, you had some.
Get a load of this, bastard. So we've got is...
When I lived with four guys, we would take turns who paid the rent each month, and so the other people would write checks to you, and then whatever the weirdest thing in the memo you could put for your money, so they had to go and deposit a bunch of
like rim job or something.
Yeah, exactly.
That'd be good.
But then the next month was going to be someone else, so they were going to get you back. You know, it was never the same person every month depositing.
You can use like Country Kitchen or something.
Yes. Yeah.
Kentucky couple.
Pause on that.
This is sort of a world record. This got world record. This was on the International Newswire.
Oh.
Cole Brower has become the first American woman to race a sailboat alone nonstop around the world.
Her husband jumped, huh?
I just want to tell you, it's really amazing, wonderful that you would come up with me on this.
Right,
I
yeah,
can't believe that I did want to go here.
I'll be right back.
I tell you, it's really a lonely out
That's
here.
really
in
just
the
a
ocean.
second.
Honey. Wait a minute, what sound did you make going in the water? That's a Don Martin thing. Cole Brower and her 40-foot sailboat traveled about 30,000 miles to compete with more than a dozen sailors in the Global Solo Challenge. Brower was the youngest and only girl in the group that set sail in October from Corona, Spain. She arrived back in Spain on Thursday, March 7th. The race took her 130 days to complete. Wow. 130 days, 30,000 miles. How many miles is that a day, Josh?
26.
What was it?
30,000 miles in 130 days.
What are you guys going to do when it's 26?
So you take 130 and divide it by 30,000. That's your problem. You're not going to have to be able to.
Yeah, it's going to be.
You got to convert it to 130 to 1,000, and then
.0043333, blah, 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 blah.
Next.
I think I'll do my new stand-up. I think the one thing we can agree on is we might not ever know how far she traveled per day.
Or you could do 30,000 divided by 130 and you get 230 miles.
We're doing math.
I know. And there are 8th graders listening that have already figured this out.
Next.
It was nicer when I said it, wasn't it?
I'm sorry, I should have told you. That's sports.
It is.
Willie literally just rubbed his heart.
It freaks me out.
Fill your own heart.
It freaks me out when I hear it, not expecting it. It's like when you drink coffee, but you think it's water, and then you get the kick. Does that make sense?
I was just hoping Jake could do it mid-sentence.
This is the new sports theme.
Really?
Let's get it going. You can keep me rolling. Josh is dancing.
You can't help it. Well, I'll twist for you, baby, and I'll rip for you, baby. Take another shot, get it for you, baby. Whoa!
I do love the oh yeah part, I tell you. Randy, you don't write checks.
How do you pay, man? If you don't write checks, how do you pay these guys?
So yeah,
Straight cash.
the screen did change. So if you stop fast forwarding because you noticed that, don't worry. You can still keep fast forwarding. I'd say probably, let's see, 36, 46, 56. About for another 20 minutes. And I know that might seem ridiculous. Like, why even have a video with this much blank space, so to speak? But it's all for a reason. One of which being I've been having issues with the recording stopping in the middle of a lecture. So I'm giving it time to stop before the lecture. That way I'm hoping if it doesn't stop between now and then, maybe it'll record the whole lecture. And if it does stop between now and then, I know there's an issue and that I need to grab my my other computer and do it that way. But anyway, yeah, keep fast forwarding.
Moss University, right?
Rand University. Yeah, and Rand is right down there where I used to work, actually.
I have a picture of me and him somewhere.
Speaking of West Virginia.
Yes, we're gonna be there.
Yeah.
Yeah, my uncle Denny called yesterday. He's gonna be at the comedy show that night. I can't wait
Uncle
to see him.
Teddy's gonna be there.
Finally get to meet Uncle
Yeah,
Denny.
I do.
So you have an Uncle Waffle House, too?
Do not make fun of her fine uncle, Denny Perkins. He's a good man. Not let you make silly puns.
We'll be there on April 5th. We'll do the show live that morning, and then we will have a comedy show that evening featuring Jeff Oskey, Josh Arnold, Willie G, Patty G, and I think your dad and I are hosting.
Fine.
What if Denny gets there and he's just drunk beyond?
No, that won't happen.
You work with my knees.
Sorry, Uncle Denny. I'm sure I'll hear about this.
She's always been a little cutie.
I like that she was.
We better take a break. The Tom Show.
Thanks for listening to the Bob and Tom Show this morning. Catch any part of the show you missed.
Texting privacy policy and terms and conditions posted at textplan.us. Texting rules for recurring automated text marketing messages. Message data rates may apply. Replace. Stop. Opt out.
Guys, got hair loss? I know what you're thinking. Should I shave my head? Comb it over? Wear a hat? Just stop. This isn't 1970. Keep your hair and your confidence, because Bosley, America's #1 hair restoration expert, can give you your real hair back permanently. Check them out today, because they're giving away an absolutely free information kit and a free gift card to anyone that texts COOL to 200-300. Dude, you don't have to look like your dad, because this isn't your dad's hair loss treatment. People all over the country trust Bosley because they're ahead of the curve. They use the latest technology to give you your real hair back. And the best part, Bosley's permanent solution is protected by the Bosley Guarantee. Let Bosley show you for free how awesome your hair could look with an absolutely free information kit and a gift card for $250 off. Text COOL to 200-300. That's COOL to 2.
Morning sunshine.
Bob and Tom Radio.
Good morning. I'm Mark Allison.
Have goods around here. Tom Griswold on assignment. That's right. He's taking the day off. That's why it sounds like things are getting a little out of hand here. Don't worry, though. Chick McGee in control as we roll through a Friday morning. Willie Griswold has stopped by as well. We're going to visit with Kevin Nealon from Saturday Night Live and Jeff Foskey with the news that we failed to mention on a Friday right here on Bob and Tom Radio.
Hi, everybody. Christy Lee with your Bob and Tom Entertainment News Update. People running the Oscars say they've got some big live moments planned for Sunday's broadcast. Executive producer Katie Milan says they've got cameos and surprises that haven't been announced. She says the Oscars are one of those last giant tentpole pop culture moments for folks to gather around the TV and watch together. The Oscars start an hour earlier this year, and that's after the daylight savings time changeover. Don't forget about that. The Oscars start 7 p.m. Eastern Sunday and will be live on ABC. Helen Mirren, who was the narrator for the Barbie movie, now has her own Barbie doll. Mattel has recreated Mirren's look on the red carpet at the Cannes Film Festival last year with a cornflower blue gown, a blue fan, and blue hair that matched the outfit. Mirren says the part that is absolutely perfect is that the doll also carries an Oscar. Mirren says having a Barbie that looks like her is one of her top achievements. And Ben Folds will once again accept requests by Paper Airplane. Years ago, Folds would ask fans to fold the request into paper airplanes and toss them for his encore. Folds says the last time he did it on tour, he got literally hundreds of paper airplanes with song requests being launched on cue. He calls it the purest, most low-tech form of engagement. Folds' Paper Airplane Request Tour will start May 30th in Charleston, South Carolina. And that's your news. I'm Christy Lee. More of the Bob and Tom Show coming up.
Ladies and gentlemen, Bob and Tom.
That's one of my favorites.
The human cannonball?
Remember the one that couple
Yesterday.
years ago, the guy in England missed the NASA?
Yeah, he killed him.
Eddie
This
Fox.
is a British man, and they to identify his body, they needed his dental records, but since he was British, there were none.
You knew
That's
he
like
would
the
love
funniest
that.
I knew exactly who my audience was.
You were talking right to him.
Bob and Tom in the morning and highlights all day long.
The Bob and Tom Show, weekday mornings at 6 a.m. Eastern on Bob and Tom 24/7.
Hi, this is Nick Griffin, and you're listening to Bob and Tom Radio.
There are some things you don't want to say to a naked guy. There are things you never say when a man is nude.
I didn't mean to laugh.
There are some things you don't want to say to a naked guy.
Can you help me find it?
Good morning. Just to give a little extra credit, since you're the first person here, and since online people don't usually get the little bit of extra credit, let's say the first word for attendance for you is going to be microphone.
Okay.
All right. And that, like I said, that will be a little extra credit. So Burton, for you, this is nothing new, but you know, like I mentioned before, we're switching. I'm still doing this transition from 101 being a 100% online course to this idea of 101 being mostly online, but all the assignments, 101 being in person, but all the assignments being online. So again, Burton, I know you've seen this, but for the rest of you, anybody who happens to be looking right now, this is what some of the questions are going to look like. I'm going to mention this again once the whole class gets here, but yeah, you can see this is what the whole some of these questions are going to look like. And what you're going to want to do when you see a question like this on the exam is just like in your brain, just completely ignore everything in black, right? Just read what's in red. As a matter of fact, if you really want to save time, you don't even need to read the first part in red. You can just jump back. You can jump straight to the second part that's in red. I know this might seem weird. It's all for a reason. If anybody wants to know the reason, I can tell you in person, but I don't want to really record it on a video or record it anywhere. So yeah, let me know. Anyway, we got some people here early, really early. So let's give an attendance word. This will be extra credit. So when you write it, when you send in your words, if I don't give you the extra credit, feel free to question me. But anyway, this word is going to be, I don't know, let's just do extra credit will be the first word. Well, will be this word for the first word for you guys. Other than Burton, who was here first. So she has 2 words, so she'll get a little bit more extra credit. But anyway, yeah. Morning. I'm just putting some final touches on this exam. Actually, there's no such thing as final in my courses. I'm putting some last minute touches to this exam. I just didn't want you to think I'm being rude, just sitting here staring at my screen when you're here. Good morning. Let's do another extra credit word since we have 2 people here and more people online. Again, these are extra credit. Well, for you, obviously they're going to be extra credit anyway, for those of you who are here in person. But for you online, these will be extra credit. Let's see, the next word will be tissue. Like something you wipe your nose with, or, a bunch of cells make up tissue. Tissues make up organelles. Organelles make up, wow. I was being recorded making that mistake. Anyway, cells make up tissue. Tissues make up organs. Organs make up organ systems. Organ systems make up organisms, so on and so forth. All right. Let's start some attendance before I project the screen here. Where you at? Oh, come on. Let's see. So who do we have online? Burton's online. Of course, not that it really matters, because when we end the call, Google sends me a spreadsheet. And then obviously, as you guys know by now, you're supposed to send me attendance/participation word. So this is me taking attendance for those online is kind of like a triple check, like a plan, not a plan B, but a plan C. If everything else goes wrong, I'm doing this. And it's being recorded. So I've got, I go up to a plan D. Anyway, Durgan is online. Of course, if you're online and wearing red, can't really tell. Plus, you've got to be present to get those free points. What else? Who else, I should say? Caleb's online. Larissa is online. I'm hoping you guys are online because spring break is starting and you're about to go somewhere fun. I hope. I was an old man by the time I started school, so spring break just meant I got to stay at home for a week and do what my wife said, or go to my job and work a full 40-hour week instead of part-time. Christopher Crowder is online. Melton is online. The other Melton. 3 others are online. That's literally what it says, 3 others, but I'll get you when you send me the words. Morning. So let's see. Morning. Is Kincaid? Yep. Kincaid is, yes.
The exam, we probably already said this. We do take that, like, we can take it over spring break.
Yeah.
Okay.
Yeah. Good question. So Kincaid is here. Abigail is here. And Kennedy, right?
Yes.
Kennedy is here. Cook is here. All right. Start projecting my screen here. So for those of you online, if you didn't hear the question, your classmate was just clarifying that, yes, you can take the exam. You have 2 weeks to take the exam. Starting from today at 9 a.m., the exam will be open. I finally fixed it. It'll be open the way it usually is via Google, or excuse me, via My Online. So you'll find the exam there. It'll be open at 9 a.m. You have 2 weeks to do it. So the Friday after spring break is when it's due at 9 a.m. Once you open it, you'll have 50 minutes to take it. Let me show you some other stuff. There is one new thing, and I want to make sure you guys don't get confused by this. If you've had my 108 class, you're familiar with this because you've seen my online exams. But I made a slight change. And once the projector comes up, I'll show you. Slight change to the exam. Once this thing gets working, I can show you. There it is. All right. So a lot of the questions, or some of the questions, are going to look like this. Whenever you see a question that's got a bunch of stuff on it, and you see black and red, just focus on the red. Don't even worry about the black. It's not there to confuse you. It's there to confuse other things that I won't even get into. So yeah, when you see a question like this, you're going to see a bunch of black with a little bit of red. The first thing in red is just going to tell you ignore everything in black. Only read the stuff in red. So if you want to save time, just go straight to the second chunk of red words. That make sense? All right. And like I said, it's all for a reason. I will point out too, some of you have already done the exam. That's awesome. So they didn't even do this I mean, the questions are the same. So they didn't do this version, and that the version they did was Google Docs. So it's the same questions, just a different way of answering them. So good job on those of you who have done it early. That was not required. And now you can relax for the next, well, relax over spring break, right? All right, let's do some more attendance. Speaking of attendance, again, I said this on Friday, but please, if you guys have friends in here that aren't attending, please let them know. Like, there's some people who, it stinks. Like, I hate to see it at the end of the semester where somebody who maybe did decent, there's a couple of people who are doing decent other than attendance, and you hate to see their grade drop just because they haven't been coming in or watching the videos. And then, of course, there's some people who are doing horrible, not just because they have bad attendance grade, but their bad attendance has also led to poor performance on assignments and exams. So please contact your classmates. Anyway, Andrea is, well, oh yeah, shoot, it's 8:01. Good grief, sorry. So if you're online and you hear me say your name and say you're not here, don't worry about it. Just send me the words as you usually do. So Andrea is not here. Let me start from the top since we're recording. Abigail is here. Kincaid is here. Andrea is not here. Mullins is not here. Lewis is not here. Thank you. If you're online, don't worry about it. I'm just going to go ahead and say, when I say you're not here, I mean you're not physically here. So yeah, I'll know you're online once you send me the words. Hunter is not here. Melton is online. Crowder was online when I checked to see who was online. Reinhardt is not here. The other Melton was online when I checked. Julia is not physically here. Kennedy is. Talbert, not here. Hannah, I remember seeing online. Larissa's online. Madeline is here in person. Hughes, not here. Galloway is not here in person. Cook is here in person. Morgan is online. Spade, not here. Burton is online. Kinder is here in person. Jones, not here. Canterbury is here in person. Hobson is here in person. Fisher is not here in person. So, okay, that's it. Is there anybody in person? No, I've got everybody in person, so we're good. Let's get this started. So again, to remind you, some of you came in after I said this, but when you see a question like this on the exam, just focus in on that second red sentence. Because the first red sentence is just telling you to ignore everything in black. The second red sentence is the question you need to answer. The black stuff is not a hint. There's no information there. It's just there to confuse a certain thing that I don't want to talk about. So again, just focus on that second red question. Zone out everything in black, and that'll help you. So, and again, the exam will be open today at 9 a.m. on Google. I keep wanting to say that. It'll be available at 9 a.m. on WVSU all night, and you'll have 2 weeks to complete it. But not that you can open it and take 2 weeks to take it, but you have a 2-week window in which you can open it and take 50 minutes to finish the exam. So any questions about the exam? All right. Once you take this exam, you are no longer allowed to retake the first exam. Some of you haven't retaken the first exam. I don't get why. Some of you I do get why, but if you failed the first exam, I don't know why you wouldn't retake it. To remind you, there's nothing to lose. If for some reason you retake the exam and this time it's not timed and this time you can use your book, and if some miracle happens, and like a bad miracle, and somehow you get a lower score, then we'll just keep your higher score, right? So it does not hurt you to retake the first exam. Plus, the questions from the first exam will be used to write the final. So not only will it help your grade, but it helps you prepare for the final. What else to say about exams? I think that's it. Oh yeah, study guides. The study guide's already there. Again, you have 2 weeks until you have to take this. So if you want to meet online to go over the study guides together as an exam review, then I'm fine with doing that. Just let me know. Yeah. And then once this exam closes and no one else is allowed to retake the first exam, at that point, you can meet me online and we can go over your exam. And I can tell you all the correct answers and all the incorrect answers. And I can tell you why they're incorrect. Because again, you need to know the correct answers for the first exam, because that's what I'm going to use to write the final exam. So just because we're done with the first exam doesn't mean we're done with it. Does that make any sense? All right. Anyway, let's get into it. Our last lecture before spring break. How exciting. Is anybody going anywhere? Where are you going? Florida? Where in Florida? That's where my friend is, actually. My friend from Tennessee is in Florida right now. Tampa. I've lived all over Florida, but never in Tampa. Anyway, this is where we left off, right? We were just like, this is the first overview. We haven't really started getting into meiosis yet, but I did show you this picture that was an overview, and I was pointing out like how, for the most part, it's a lot like mitosis with the differences that we already talked about on Wednesday. So you can see here, the parts that are the same are in prophase, the nuclear envelope has disappeared. The spindles are starting to move so that they can attach to the chromosomes. Then they start moving towards the middle of the cell. Once they get to the middle of the cell, that's metaphase, just like it is in mitosis. Once they're in metaphase, it's kind of like a tug of war trying to pull them apart. Once they do separate, they're considered full-blown chromosomes again, and that is anaphase, right? And then they keep moving apart towards the opposite ends of the cell. Once they hit the opposite ends of the cell, that is considered telophase, at which point, and you don't see it in this picture, but the nuclear envelope reforms, the spindle disappears, which they do show, cytokinesis happens, and that one big cell turns into 2 cells, right? So that looks very similar. The only difference is, at least by looking in this picture, the big difference is we're dealing with homologous chromosomes for the first round of meiosis, not sister chromatids, right? So you can see here at the end of it, we still have sister chromatids attached, right? We haven't separated them like we did in mitosis, right? This is only the first round of meiosis. So the only thing we've separated is homologous chromosomes. And then, of course, like I mentioned on Wednesday too, one of the other things that makes meiosis different than mitosis is this right here, crossing over, right? So these homologous chromosomes, the one that you got from your mom, the ones that you got from the dad. Sorry, I heard a noise. I'm going to check on that. Anyway, the homologous chromosomes, which are the same ones, one from your mom, one from your dad, they swap chunks, right? So you have a mom chromosome with a dad chunk on it and a dad chromosome with a mom chunk on it. Let's see what that noise was. Can you guys, maybe they can't hear me. That's unfortunate. Can someone confirm they can hear me?
We can hear you. It's just the slide. We can't see it.
Oh yeah, thank you. I'll fix that. Yeah, the camera's still messed up. I got it to at least record, but I can't get it to zoom in or out, and I couldn't even get it to aim at the screen, so I had to go up there and manually move it earlier today. But anyway, thank you for letting me know. Let's get back to it. All right, so again, that's just the overview of the first round of meiosis. And remember, there's 2 rounds of meiosis. So everything I just said, here it is in writing. Prophase 1 is essentially the same as mitosis, except we're not dealing with sister chromatids. We are dealing with homologous chromosomes. So for this portion of the exam, you should automatically associate prophase 1 with homologous chromosomes, or vice versa, homologous chromosomes with prophase 1. Of course, later in other chapters, we're going to talk about homologous chromosomes in different contexts. But when it comes to mitosis or meiosis, when you think of homologous chromosomes, you should think of meiosis 1. And in this particular slide, specifically, prophase 1. So the pairing of these homologous chromosomes is something that I put on the screen, but I'm not going to ask you, but the book includes it, so did I. It's called synapsis. But I'm not going to ask you that, so let me put an X through there. But your first official word for attendance will be pair. Anyway, yeah, it's called synapsis. And that's a word I don't think I'm going to use it for the rest of this chapter. And I know I'm not going to use it after this chapter. So again, that's why for me, it's an unimportant question. Of course, I haven't read the textbook in a little bit, so the textbook might refer back to it a few times. So just in case, now you know. So when you're studying your textbook and they use that word, you know what it means, right? So remember, to put it in some sort of different context, if you have one chromosome and it duplicates itself in S phase, and now they're attached, you've got 2 of them and they're attached to the centromere, right? Those are sister chromatids. Now, when you bring 2 homologous chromosomes together, those aren't sister chromatids, but that's called a synapsis, right? When you have 2 of them together, it's called a synapsis. And also, like we mentioned earlier, crossing over occurs. That's very important, right? That gives us our sexual selection, which will be, or excuse me, how do I put that? A lot of what drives evolution is this variety, right? We have variety, and we get variety from sexual reproduction. And part of that variety from sexual reproduction comes from crossing over. So again, every single chromosome in your body, you got one from your mom, one from your dad. But because of crossing over, some of your mom chromosomes have a chunk of dad on them, and some of your dad chromosomes have a chunk of mom. That'll be really important later when we start talking about genetics. Anyway, again, your book, let me say this again, it's a good time to remind you. For the exam, the way I teach it, I teach an easier version. So just like in the lab, meiosis and mitosis, the phases are prophase, metaphase, anaphase, telophase. And like I mentioned earlier in the week, or possibly last week, your book gets a little bit more complicated and dives deeper. And they differentiate between prophase and prometaphase. I do not. So read about it if you want. But just remember, for the exam, I'm keeping things simple. And prophase and prometaphase, as far as we are concerned, it's the same thing. So any questions about that? All right. There it is again. I'm not going to talk about this picture. Well, no, let me see. So this is meiosis 2. So we've already gone through meiosis 1. We just talked about it, right? So in there, the homologous chromosomes came together. They met in the middle. They got pulled apart in anaphase. Then they were all the way apart in telophase. Then cytokinesis happened and it pinched off, and we had 2 cells. Well, now those 2 cells are here, and they are going into meiosis 2, right? So we have 2 cells now instead of 1, but you could just focus on 1 because they're doing the same thing at the same time in this picture, right? So the same thing's happening, right? The nuclear envelope is gone. The spindles are moving, and they're going to attach to the sister chromatids, right? We're not dealing with homologous chromosomes anymore. They attach to the sister chromatids, which you can't see here, and then they start moving towards the middle of the cell. And then once they get to the middle of the cell, that's metaphase, right? And now there's a tug of war trying to pull those sister chromatids apart. And then finally, once they break, that is anaphase, right? Now they are full-blown chromosomes. They're no longer sister chromatids because they're not attached. They're moving towards the opposite ends of the cell. Once they get to the opposite ends of the cell, that is called telophase, at which point the spindles detach, which makes sense because we don't need to move the chromosomes anymore. The nuclear envelope reappears, which you can't see in this picture. The chromosomes uncoil, which you cannot see in this picture. But yeah, there you go. So again, the big difference between meiosis and mitosis. In mitosis, we're only dealing with sister chromatids. In meiosis, at first we deal with homologous chromosomes, and then in the second round we deal with sister chromatids. So homologous chromosomes in the first round, sister chromatids in the second round. Also, that's another thing worth mentioning is that the second round of meiosis is essentially, I mean, with some differences, but between the first round of meiosis and the second round of meiosis, the second round of meiosis is the most like mitosis. Because again, we're dealing with sister chromatids. Some other differences to note, and I'll probably have a slide for this later, but I'll go ahead and say it now and then show you later. So remember in mitosis, we started with one cell, we ended with two. In meiosis, which is what we're talking about now, we started with one, and then after the first round, we ended with two. And then after the second round, we ended with 1, 2, 3, 4, right? So we have 4 genetically unique cells as opposed to 2 genetically identical cells. Any questions about this slide? Oh, good grief. I apologize. This slide should not have been there. That is meiosis 2. We were talking about prophase 1, and then I jumped into meiosis 2, and I thought that was odd, and I thought maybe I did that on purpose, but I didn't. That was a mistake. So I apologize. Anyway, back to prophase or metaphase 1. So we're going back to talk about the first round of meiosis. I apologize. Anyway, it's essentially the same as mitosis. Again, this is metaphase 1, the first round. So we're dealing with homologous chromosomes that are meeting in the middle, not sister chromatids. Here's another thing. This is going to be really important later. For now, I'm just going to say it's important. I'm going to show you this word. Later, I'm going to define it. Later, it's going to be important. And historically, I think it's safe to say that'll be complicated. People seem to have an issue with independent assortment. But for now, I'll just explain it like this. It's not like, you know, when your chromosomes line up in the middle, or excuse me, homologous chromosomes line up in the middle, it's not as if these are all mom chromosomes on one side and dad chromosomes on the other. It's completely random. It's all independent. So this one could be mom and dad. This one could also be mom and dad, one mom, one dad. And this one might be, I don't know, dad and mom. But imagine doing that 23 times, right? Because you have 23 pairs of chromosomes. So those 23 pairs of chromosomes can line up any which way. And that'll be important, very important later. But for now, I'll just let you know that that's what it's called. That's what independent assortment is. And later, it'll be really important. Anyway, now we're going to talk about anaphase and telophase of the first round of meiosis. So again, we're still talking about the first round of meiosis. I had to back it up. So again, we're still dealing with homologous chromosomes, not sister chromatids. So it's the homologous chromosomes getting pulled apart. Yeah, that's it. That's the only thing we say about anaphase 1 and telophase 1. So that means we're now officially done with talking about the first round of meiosis, excuse me. So let's talk about what I've already talked about and jump back into meiosis 2. Again, meiosis 2 is essentially the same as mitosis, right? This is of the 2 rounds of meiosis, this one is the most like mitosis. The differences are the end product cells, which I already mentioned earlier. Well, I didn't mention this actually. So in mitosis for a human, you start with a diploid cell, you end with 2 diploid cells. For meiosis, you start with a diploid cell, which means it has all the chromosomes, the full set, and you end with haploid cells, which again, that means it has half the number of chromosomes, right? So if you were to look at any cell in your body, except for your sperm or egg, again, you would have a chromosome, 1 from each parent. Now, if you were to look at your sex cells, your sperm, your eggs, your gametes, and look at the chromosomes, there's only 1 of each, right? So chromosomes 1 through 23, you only have 1 of each. Could be from your mom, could be from your dad. Statistically, this would be very rare, but statistically, you might have a cell that has all mom chromosomes in it. And statistically, you could technically have a cell that has all dad chromosomes in it. But more than likely, it's going to be some combination. And later, I'm going to tell you about the number of combinations there are. To me, it's mind-blowing. Anyway, The other big difference is that, again, we end with 4 cells in meiosis, 2 cells in mitosis. And again, in meiosis, they are genetically unique. The cells are not identical. They're unique to each other and to the parent cell. Unlike mitosis, which is what we learned about earlier in the week. And in mitosis, you start with 1 cell and then with 2 genetically identical cells. So, any questions about meiosis 2? Okay. The next word for attendance is 4. And the word after this, I don't know what it's going to be yet, but I'll go ahead and tell you now. I'm not going to say it. I'm just going to circle it. So be aware for a circle. Anyway, there's meiosis 2. I've already shown you this picture on accident. I showed it to you early, so there's no need to talk about it again. So let's talk about comparing, again, the big overview. Comparing mitosis, meiosis. This is a good time for me to remind you, this is going to be on the 3rd exam, so not the exam that you have to take today, or starting today. And most of the exam for this chapter is going to be about mitosis. And most of the questions about meiosis are going to be more like comparing and contrasting. Like, how is meiosis different than mitosis? How is meiosis the same as mitosis, right? So there'll be a lot of comparing and contrasting when it comes to the meiosis questions. But that being said, let's have the review here. For both meiosis and mitosis, the chromosomes only duplicate that one time in interphase, right? So it goes through interphase, the chromosomes double, right? That only happens once, whether we're talking about meiosis or mitosis. In mitosis, we have the 1 division, right? So it doubles, then it divides once, right? So it cuts it in half. But once you cut it in half, that goes back to the original number, right? So again, let's say you start with 3, then you double it up to 6, you cut that in half, and then you're back down to 3, right? In meiosis, even though we only have the 1 duplication, we're dividing twice, and that's how we end up with 4 haploid cells, right? You take 1 cell, you divide it in half, you get 2 cells. You divide those in half, you get 4 cells, right? So again, big difference here. Meiosis, you end up with 1, you start with 1, you end with 2. Meiosis, you start with 1, you end with 4. Mitosis ends with diploid cells. Meiosis ends with haploid cells, right? There's big differences. The events that are unique to meiosis only occur in meiosis 1. Another way of saying that first bullet point is meiosis 2 is essentially the same as mitosis. There are some differences, but mostly it's the same. Because again, for meiosis 2, we're separating sister chromatids just like we did in mitosis. Unlike mitosis, meiosis 2 yields haploid daughter cells. So I keep saying that. You keep seeing it on the screen. Hopefully you remember. So there we go. You can see the difference between mitosis and meiosis here in one picture, right? You can see the chromosomes, if they duplicated, line up in the middle, they get separated, right? Here, chromosomes are duplicated, line up in the middle, they get separated. You end up with 4 cells instead of 2. All the stuff I already told you, there it is again. And these pictures are not from your textbook, just so you know. I mean, there's pictures like this in your textbook, but I wanted to show you something a little bit different. So that brings us to this. This is a really, really important bullet point because this information that you're going to get now is going to carry on chapters from now. A lot of this other stuff, meiosis and mitosis, it is important. You do need to know it. But if for some reason that's not your cup of tea and you're struggling with it, you should be okay once we move past this because we're not really going to go back to it too much. But this, the origins of genetic variation, this will be very important when we talk about genetics and when we talk about, even DNA and when we talk about evolution. So here we go. The offspring of sexual reproduction are genetically different from the parents and one another. I've already said it. We're saying it again. Of course, what I've said previously, most recently, is I was saying that from a cellular point of view, right? So the 4 cells are genetically unique from each other and to the parent cell. But then the same can be said for the whole offspring, right? So just using humans as an example, since we are humans, right? When 2 humans get together, make babies, Unless they're twins, all those babies are genetically unique from each other and genetically unique from the parents. So, how does meiosis produce this genetic variation? I'm going to go through this not quickly, but not really slow either. I'm going to kind of push through it because even though this is a really important concept, we are going to come back to it at a later chapter, and we're going to dig into it deeper at that point. Plus, a lot of what I'm about to say is kind of a repeat of what I've said earlier today. So let's talk about it. Again, independent assortment. In metaphase 1, the side-by-side orientation of each homologous pair of chromosomes is by chance. So once again, it's not as if all the mom chromosomes, well, try to again picture in your mind all those chromosomes, those homologous chromosomes lined up in the middle of the cell for the first round of meiosis, right? And it's not as if all mom cells are on one cell side and all dad cells are on the other, right? They can go any which way for all 23 pairs of our chromosomes. So that means there are 8 million possible combinations just talking about how they line up. That doesn't even include crossing over. And that's only one parent. So for my sperm, statistically, if I were to look at 8 million of them, each one of them would have a different arrangement of how the chromosomes are lined up, right? So like, I got chromosome 1, I got from my mom. Chromosome 2, I got from my mom. Chromosome 3, I got from my dad. Chromosome 4, I got from my mom, right? Whatever it may be. Imagine that 23 times. 8 million possible combinations. Take that into account with the fact that, you know, to sexually reproduce, you have to hook up with somebody else, so to speak, right? So just a sperm isn't going to do it. Just an egg isn't going to do it. They've got to combine. Therefore, a man and a woman can produce 64 trillion combinations of chromosomes. This means statistically, if you could somehow get a man and a woman to have 64 trillion children, it's very feasible that they would all be genetically unique only in which chromosomes they have. Again, that doesn't even include crossing over. So even though one might have chromosome 2 from mom and chromosome 3 from dad, even though that might be the case, chromosome 2 might have a chunk of chromosome, mom's chromosome 2 might have a chunk of dad on it, right? So think about those numbers, 64 trillion. Here's what I like to think about. This is mind-blowing to me. You guys ever heard of the butterfly effect? Or ever seen like a time travel movie where like, be careful when you go back in the past. One slight change will change everything, right? So think about this. And this is kind of weird. And I'm not going to test you on it, but I think about weird things like this. 64 trillion possibilities, right? So to make you, your mom and your dad, assuming it was normal fertilization and wasn't IVF or anything like that, even then, these numbers are still relevant. Your mom and your dad had to have intercourse and that exact way, at that exact time, and orgasm in that exact position at that exact time for you to exist. Any slight variation, because look, there's 64 trillion combinations. If your dad got off a minute earlier, it wouldn't be you, it would be your sibling. Isn't that crazy? And think about all the things that go into that, right? Maybe your dad was driving home, somebody cut him off that day. He was a little bit in a pissy mood. So because he was in a pissy mood, he was having intercourse, took him a little bit longer to get off. Now it's a different child. You see what I'm saying? I know that sounds a little bit vulgar, but it really is crazy to think all of that had to happen for you to exist. And then think about this. All of that individual happens for every single human to exist. So for your parents to exist, that had to happen for them. And then obviously you wouldn't be you without your parents. So the odds of us being here are just so tiny. And such a slight change really would change the whole world. If you could go back and change one tiny thing about the past, it would change the future tremendously. So yeah, think about that. That's crazy to me. I don't know. It's biology related, but it's not strictly a biology topic. Your parents had to hook up just the way they did. If it was missionary, it had to be missionary. Can't do anything else, or vice versa. Anyway, so here we go. This is what I'm talking about. So this is what I was talking about, except in this hypothetical situation, whatever organism this is, whatever species, they only have 2 chromosomes, right? So we have 23. This is dealing with 2 chromosomes, right? So the possibility number 1, let's call mom red and dad blue. Why not? So in this possibility, they happen to line up with all dad on one side, all mom on the other. Just happen to. So then those separate. So we have these two cells that are 100% dad, 100% mom. And then obviously when they separate, there's going to be 100% mom, 100% dad. Here, they're swapped. So now you have this cell that has mom chromosome and a dad chromosome. Here you have a dad chromosome, mom chromosome. And then, of course, they separate, right? So this is only with 2 pairs of chromosomes. And again, you have 23 pairs of chromosomes. Your existence is a miracle. This slight change will change everything. Anyway, so again, that's independent assortment. Crossing over, we've already talked about. So again, think about all those 64 trillion combinations that just dictates which chromosomes you got, whether it be from your mom or your dad. And then consider the crossing over, right? Crossing over means, yeah, you might have this chromosome from your mom and this from your dad, but that mom chromosome might have a chunk or 2 or none from your dad and vice versa. So what is crossing over? Like we've already said, here it is in writing again. You need to know what it is. It is the exchange of corresponding segments between homologous chromosomes. Like I've already said, it happens in prophase 1. Which makes sense too, right? So if you were to get that question on the exam, if you were to say, I don't know, metaphase 1, you'll lose points, but you'll get partial credit. If you were to say something like prophase 2, you'll get 0 credit. Because the idea here is, remember, in the first round of meiosis, the homologous chromosomes are together. And the second round of meiosis, they're separated. So there's no way you can have crossing over when the homologous chromosomes aren't even together. So keep that in mind. Also, just to let you know, to remind you for this exam you have coming up, exam 2, there are some questions like that. Again, if you have a question about photosynthesis and you answer incorrectly, but your incorrect answer was a photosynthesis answer, you'll probably get at least a little bit of credit. If it's asking you about photosynthesis and you answer something about respiration or mitosis or something, you're going to get 0 credit. So I hope that helped you. Anyway, so that's crossing over, right? We already talked about it. We know what it is. Let's look at this next slide. That's what it looks like, right? And again, like I mentioned, first of all, crossing over doesn't always happen. The first time I showed it to you, there was only like 1 chunk. This picture is showing 2 chunks. Sometimes the chunks are smaller. Sometimes the chunks are bigger, right? So there's so many variations that could happen, right? It's all very random. And again, this is crossing over. Add this randomness to the 64 trillion thing that we talked about earlier. Again, you are a miracle. I mean, you wouldn't be you if anything went different. Anyway, let's talk about what happens when meiosis goes wrong. This is slightly important. That's the next attendance word. So what happens when there are errors in meiosis? Well, first of all, what are errors in meiosis? Well, let's think about what meiosis does. Meiosis first separates homologous chromosomes. Meiosis then separates sister chromatids. So when we say errors in meiosis, that's what we're talking about, right? Something's not separating, right? So either homologous chromosomes aren't separating or sister chromatids aren't separating, depending on when it happens. And when that happens, this can result in genetic abnormalities that range from mild to severe to fatal. How can you find out if you have them? Well, with a karyotype, like I mentioned earlier in this chapter. The karyotype is basically just a picture of your chromosomes, right? They used a microscope. They got your chromosomes that were lined up in metaphase, so you can see them. They dyed them. They took a picture. You can see them. They cut and paste it and range it. That's a karyotype, right? So that is one of the things you can diagnose with a karyotype. You can diagnose when there was errors in meiosis, because at the end of the day, when there's an error, it's either going to be too many chromosomes or not enough. And that is something you can see with a karyotype. Any questions about this slide? All right. Moving right along. Maybe we can finish this chapter. That'd be great. Let's talk about how these accidents can alter the chromosome numbers, right? The word for it is non-disjunction. Let me put a question mark next to that because I'm not sure I'm going to ask you the name of it. You definitely need to know what I'm about to teach you. I'm not sure I'm going to test you on the name, but anyway. Non-disjunction is when the chromosomes fail to separate. Of course, at anaphase, I copied and pasted from your textbook. So your textbook says non-disjunction is when the chromosome pair fails to separate at anaphase. And I'll go ahead and put a line right here. I'll tell you right now, what you need to know if you're taking notes or making note cards to study, you could just leave it at that. Chromosome pairs fail to separate. I'm not going to add at anaphase. I just assume you know that, right? That's when all chromosomes separate is during anaphase. And I'm already going to ask you a question about anaphase, so I don't want this question to be about anaphase. So again, the multiple choice questions are not going to be non-disjunction is what? Chromosomes pairs fail to separate at anaphase, at metaphase, at telophase, right? I'm leaving that out. Non-disjunction is when they fail to separate. So what happens when they fail to separate? Well, then you get sperm and egg or gametes that don't have the right number of chromosomes. So they can have too many chromosomes. They can have not enough chromosomes. When I put this in red, because this is probably the most important thing on the slide as far as the exam is concerned, non-disjunction can happen in either round of meiosis, meiosis 1 or meiosis 2. Meaning, if it happens in meiosis 1, it's the homologous chromosomes that fail to separate correctly. If it happens in meiosis 2, the sister chromatids that fail to separate correctly. Notice too, I haven't mentioned anything. We had the whole conversation about mitosis without mentioning this. So for independent work, if you want, you could look that up. What happens when what happens when you have non-disjunction in mitosis, right? Anyway, any questions about this slide before we talk a little bit more about non-disjunction? A visualization of what's on the slide right now? All right. Well, I guess that'll be the next slide. When you have the correct number of chromosomes, we call that euploid. When you have the incorrect number, we call that aneuploid. And that includes two different types, which is monosomy, which is the loss of one chromosome, or trisomy, which is the gain of an extra chromosome. I'm going to put a big old question mark through this because that's indicating most likely I will not ask you anything on the slide. We will talk a little bit more about trisomy, but probably I'm not going to ask you any of this. And if I did, maybe it'll be an extra credit question. Anyway, any questions about that? It's just a bunch of naming stuff, right? Euploid, you're normal. Aneuploid, you're not normal. You either have too many chromosomes or not enough. And again, for extra credit, if you want to look this up, and I don't even know this, I'm just guessing here, but what happens if you lose more than one chromosome or gain more than one? I don't know this, but I would assume that is such a big change that the fetus or the zygote wouldn't even survive. Having that many chromosome abnormalities, I think that would just end up in a miscarriage, I would assume. Anyway, any questions about this slide before we have the visualization? Okay, there's the visualization. So on the left, we have the non-disjunction happening in meiosis 1, right? So that means it's the homologous chromosomes that are failing to separate. That's bad, right? That means everything's going to be messed up, right? So here, this thing finally separates. So this cell has too many chromosomes. This one has not enough. That separates. So again, this one has too many cells. Or excuse me, too many chromosomes. This one has too many chromosomes. This one doesn't have enough. This one doesn't have enough. Here, we can see what happens when it happens in meiosis 2. So we're talking about sister chromatids here as opposed to homologous chromosomes. Because the homologous chromosomes separate as they should. But then here, these sister chromatids fail to separate. This one did it just fine. So in this situation, we at least have two good gametes, right? Two good sperm or egg. Here, we have one that has too many chromosomes and one that has not enough. So there you go. You might get some question. Let me think. How am I going to word this? You definitely don't need to memorize, like, for example, meiosis 1, non-disjunction of meiosis 1 leads to n plus 1, n plus 1, n minus 1, n minus 1. Meanwhile, non-disjunction of meiosis 2 leads to n plus 1, n minus 1, n. I'm not going to ask you that necessarily. I don't know how I would ask it. Maybe like, which one would be more harmful? Non-disjunction of meiosis 1 versus non-disjunction of meiosis 2. And what's your answer to that? Take a look at the board. Which one's worse? Non-disjunction in the first meiosis on the left or non-disjunction in the second meiosis on the right? Which one's worse? Don't overthink it. That's probably you're looking. I'm hoping you're looking at this and like, surely it's not that simple. I don't want to spit out the answer and look dumb. So it is that simple. Which one's worse, left or right? The one on the right, yeah. Excuse me. The one on the right is better. The one on the left is worse. Look, too many chromosomes. Too many chromosomes. Not enough chromosomes. Not enough chromosomes. Too many chromosomes. Not enough chromosomes. Perfect. Perfect. So yeah, I guess that is a big concept thing. Make sure you know that. Not because it's necessary that the fact itself is trivial. Like which one's worse, non-disjunctional meiosis 1 or non-disjunctional meiosis 2? That's a trivial fact. But to be able to think about what non-disjunction means, then you should be able to figure out, meiosis 1 would be worse. Not because it's trivial, but because you think, all right, let's fail to separate in the first round. Therefore, we have 2 messed up cells going into the second round. Therefore, all the cells that we produce are going to be messed up. When it happens in meiosis 2, only half of your cells are going to get messed up. Anyway, any questions about that slide? And this picture is not from your textbook. So if you're trying to study and you're looking for your textbook and you're looking for that picture, it's not there. Come back to the PowerPoint. All right, the next slide I'm going to show you shows an abnormal gamete, sperm or an egg, fertilizing a normal gamete, sperm or an egg. So we have a normal cell coming together with an abnormal cell. And the result is going to be a zygote that has 2N plus 1 chromosomes. And I already told you, I'm not going to use that terminology in the exams. I'm not going to ask you anything about 2N or N plus 1, none of that. But basically what we're saying here is, the layman's terms, when this happens, it results in a zygote that has an extra chromosome. That's what that means. Mitosis then duplicates that abnormal cell, right? So again, the sperm and the egg come together, and that very first cell of life, that human being, in this case, I'm using that as an example because it could be any sexually reproducing organism. Anyway, that one, everything starts as one cell to begin with, and it splits into 2, 4, so on and so forth. But if your first cell that you begin life at is messed up, and you don't grow other than mitosis, and mitosis makes a perfect copy, then that means the rest of, all your cells are going to be messed up, right? Because you began life with the wrong amount of chromosomes, and then that one cell was perfectly copied over and over. Which also brings me to another good bullet point, not bullet point, independent work topic, depending on how deep you want to look into this. What happens if you have a normal sperm and a normal egg? They come together, you have a normal zygote, right? And then that thing splits into 2 cells. So here you have 2 normal cells, right? And then those things split, and one of those cells then goes through non-disjunction. So now you have half of your cells completely normal, and then half of your cells have too many chromosomes and not enough, right? So again, to be clear, what we're talking about here that you need to be responsible for is all the cells in your body, other than your sperm and your egg, having the wrong number of chromosomes. What I'm suggesting for independent work is, what happens if it's like half the cells in your body? Is that even possible? You have somebody that has half of their cells too many chromosomes and half normal. Just something to think about. So here's what I'm talking about. There's your normal sperm, right? Again, this organism, this species, whatever we're talking about, only has 2 chromosomes, right? 2 pairs of chromosomes. So anyway, in this case, we have normal one. That one has an extra. They come together. So there's a pair of homologous chromosomes. There's a pair of homologous chromosomes, and then there's that one extra of whatever, that bigger chromosome, there's an extra, right? Now this cell, that's your zygote, right? That's the very beginning of life. So that cell is going to be perfectly copied. And then those 2 cells will be perfectly copied. And then those 4 cells will be perfectly copied. So again, once that mistake is there, it's there. Which brings us to this next bullet point, something called Down syndrome. I'm sure a lot of you heard about it, but here we go. It's basically also called trisomy 21 because that means there are 3 number 21 chromosomes. And again, to remind you, we have a normal human has 23 pairs of chromosomes. What we're saying here is that this person does have 23 pairs of chromosomes, except for on the chromosome number 21, they don't just have a pair, they also have an extra, right? Some stuff, I'm not going to ask you these numbers, but just so you know, this affects about 1 out of 800, about 850 children. You don't need to know that, but your book included it, so did I. I might ask you this, and this is probably why your book even included it. It is the most common chromosome number abnormality. It is the most common, which is probably why you've probably heard of it. That's probably why your textbook decided to talk about it. Which brings me to another independent work topic. If you have an extra chromosome on chromosome 21, it's called trisomy 21, and it's Down syndrome. So now you have a world of opportunities of things to look into. What if you have an extra chromosome number 1? What if you have an extra chromosome number 2, number 3, number 4, all the way down, right? Of all 23 pairs, what happens if you have an extra of any of the other ones? Or what happens if you're missing any of the other ones? What's the syndrome called? Can you survive it? What are the symptoms? So many things you can look into. Anyway, just like this bullet point here, the second bullet point also points out that it is the most common serious birth defect in the United States. Again, I might ask you that. I'm not sure. But again, this is why we're mentioning it, because it's so common. Again, I guess all these things are, I might ask you. But the risk increases with the age of the mother. The 35 years and older, they're at higher risk. And you could look into that, right? Look into that if you want. What is it about older women that put them at higher risk to have extra chromosomes? Why not older men? And what exactly is happening at the cellular level? Like what happens when you're old to where you're starting to get non-disjunction? Like in my mind, and we don't even talk about this, of course, what happens to you? Well, you have chromosomes, as you know, we have talked about that. And what happens is little tips of your chromosomes, they're called telomeres. And they're there to protect the good stuff in the middle. So the tip of your chromosomes, they get a little bit chopped off every time they reproduce, which is one of the reasons we age, right? So every time it gets duplicated, there's a little bit that gets messed up. But in my mind, that wouldn't cause non-disjunction. So in my mind, I can see how getting older, you have older cells, and there might be some other problems, but not that one. So look that up for independent work if you want. Anyway, any questions so far about trisomy 21? I'm definitely not going to ask you this, but again, your book included it, so did I. People with Down syndrome have characteristic facial features, generally speaking, and I know you're not supposed to judge a book by its cover, and I don't mean this in any bad way, but I think, generally speaking, if you were to show me somebody with Down syndrome, like, yeah, they have Down syndrome. I'm sure you've seen them. They have a particular look. There's actually your textbook included a picture. I'll share that later. They generally have a shorter lifespan on average. There's varying degrees of developmental delays, so it's not all equal across the board. Not everybody who has trisomy 21 or Down syndrome, they're not exactly, their affliction isn't the same, which is this bullet point right here. Some may live to middle age or beyond, and some may be socially adept. To be clear, because this seems almost like an offensive slide in some ways, I'm teaching out of the textbook. This is from the textbook, copy, paste, make some adjustments like you shouldn't do in independent work, but it's okay when you're making a PowerPoint from a textbook. Yeah. So if that offends you, take it up with your textbook authors. Anyway, here's the karyotype. This is a normal karyotype, right? There's 2 of each chromosome, right? 2 of number 1, 2 of number 2, so on and so forth. You can see here, trisomy 21 looks completely normal. But then here's trisomy 21. This one, instead of having 21 chromosomes, there's only 3. And again, to remind you, a good independent work topic would be, what happens if you have an extra chromosome number 1 or 2 or 3, so on and so forth. I have a hypothesis that I still haven't looked up. Why is trisomy 21 so common? That's the question. My hypothesis is because the chromosome is so small. Because remember, chromosomes are strands of DNA that have instructions on how to build proteins. So literally, the bigger it is, the more genes there are. Does that make sense? Because there's literally more DNA. The smaller they are, the fewer genes there are, because it's just a smaller chunk of DNA. So in my mind, it makes sense that you can have an extra number 21 because it's so small. It's not making much of a difference. In my mind, if you have an extra chromosome number 1, it seems like it'd be a bigger deal, right? That's a huge chromosome. So you can look that up for independent work if you want. Again, that's just my hypothesis. Same way with this. The sex chromosomes. Is it? Yeah, the sex chromosomes are also pretty small. Well, is that the sex chromosomes? Yeah, that's a female. Anyway, the sex chromosomes are not as big as the number 1, for example, but they are kind of big. And we'll talk about what happens when you have too many sex chromosomes later. Are there any questions about this slide? All right, the next word for attendance will be baby. Phew, we might be able to do this. The last bullet point of the PowerPoint is abnormal numbers of sex chromosomes, like I was alluding to, right? So non-disjunction can lead to abnormal numbers of sex chromosomes, just like any other chromosomes, right? Number 1s through 23. Now we're talking, or number 1 through 22. Now we're talking about that 23rd pair, which is the sex chromosome, right? Your X and your Y if you're a male, X and X if you're a female. This upsets the genetic balance less than autosomes. So for some reason, when you have an extra number or not enough of a particular sex chromosome, it's usually less impactful than the other ones. This is, I think, from your last textbook. You do not need to know this, anything about this. The point is that you can have extra X chromosomes, you can have extra Y chromosomes, you can have some missing. They all have a name. We have statistics as how common they are, and you can see what caused it, and you can see the symptoms. But you do not need to know any of that. If anything, maybe I'll throw in some questions about that in the extra credit section, like, what do you call XYY? Well, it doesn't have a name. Wait a minute. Slightly taller than, okay. Let me say this. I'll leave it with this. This is good for independent work. Any criminal justice majors in here? No? Anyway, I've read something, and I keep saying this every semester, and I've yet to look it up. But the prison population, I've heard, has a higher percentage of males that have an extra Y chromosome. And the Y chromosome is what makes you a male. And I think the hypothesis is because they have that extra Y chromosome, they have extra testosterone, which makes them extra aggressive, which makes them a little bit more likely to do violent crimes. So anybody can look that up if you want for independent work. You know what? Yeah. No, we can finish. I'm so torn. Let's talk about this really quickly. The damaged chromosomes, we have partial duplications and deletion. You know what? Nah, I don't want to rush through this. So when we come back next week, we're going to finish off with this last slide on this chapter, and then we'll start the next chapter. And I guess the last word or thing for attendance is birthday. Today is my son's birthday. So happy birthday to Pierce. And that's it. I hope you guys have a great week. I hope to take some time off, but I will probably be working. So if you need me for anything, let me know. You too.